Proteins are made of a long chain of amino acids, which has been coded for by DNA. The order of the bases of DNA determines which protein is made by coding for a specific order of amino acids. This is protein synthesis, and in this video, we're going to look at how it works. You may want to watch our What is DNA video first to remind yourself all about DNA. A set of 3 bases in the DNA molecule codes for a particular amino acid. GGT, for example, codes for the amino acid glycine. The 3 base codes are known as codons, so GGT is a codon for glycine. So there are 20 amino acids that make up our body's proteins. 9 of these we need to take in through our diet, as our body cannot manufacture them. These are known as essential amino acids. Our body can, however, manufacture non-essential amino acids. Whilst 20 amino acids build up proteins, there are actually many others that do not form proteins, possibly over 250. They may form sugar, for example. Before we start, you need to know what RNA is. So you may want to watch our What is RNA video first. So let's now look at how proteins are actually synthesized. The DNA is in the nucleus and cannot move, but the ribosomes in the cytoplasm are where the proteins are made. This means the code from the DNA needs to be copied and carried across to the ribosome by a molecule called messenger RNA, or mRNA, before the protein can be made. Let's have a look at how all this works. In the nucleus, the enzyme RNA polymerase unwinds and unzips the 2 strands of DNA that contain the protein-making gene. Only one of these strands is going to be replicated. Complementary RNA nucleotides base pair with the chosen strand. RNA polymerase also then binds the RNA nucleotides together, making a new RNA strand. This is called messenger RNA, or mRNA, and this whole process inside the nucleus is called transcription. The mRNA then travels from the cell nucleus and out into the cytoplasm until it reaches and attaches to a ribosome. The ribosome then sticks amino acids together to make a polypeptide chain, following the order of amino acids as coded for by the mRNA. 3 base codons on the mRNA code for 1 amino acid, and this whole process in the ribosome is called translation. So let's look at translation in a little more detail. What is actually happening inside the ribosome? Transfer RNA, or tRNA molecules, carrying specific amino acids base pair with the mRNA inside the ribosome. So the tRNA brings in the amino acids, and the mRNA provides the information of the exact order that the amino acids need to be bonded together to ensure that the correct protein is made. As more and more tRNAs match up with the mRNA, the amino acid chain becomes longer and longer, and eventually the polypeptide chain will form into the protein. So there we have protein synthesis. Inside the nucleus, the DNA molecule is unzipped from around the gene by DNA polymerase. The DNA is then replicated and mRNA is formed in the process of transcription. The mRNA has to travel out of the nucleus and into the cytoplasm, where it binds with a ribosome. In the ribosome, the nucleotide sequence of the mRNA is translated by the tRNA, which carry the related amino acids. The polypeptide chain is formed and will eventually fold into the required protein. So there is protein synthesis.